Okay, so thanks for the invitation to be here. It's been a really good day so far. So as said, I'm going to talk about the strategy for large-scale multi-omics in UK Biobank coming from some of the UK Biobank meetings that we've had. So let's start off with something that's been talked about already. Rory's mentioned it, others have mentioned it, Bernard mentioned it, the business of what samples have we got and how are we going to use them. The interesting the biofluids for us are going to be plasma, urine, and indeed saliva. And as you can see from the aliquots available here, there's quite a lot of aliquots of plasma and serum available, urine, indeed even more available, and some aliquots of saliva. In terms of the amount of material needed for metabolomics, then you would not be depleting this set of samples to a very great degree to undertake metabolomic studies. And in terms of proteomics, you'd be depleting them even less. So currently, I think you can get great data on hundreds of proteins with less than 100 microliters of plasma. That's the kind of level we're talking about. So we know that we can do it, and it wouldn't deplete the samples to too great a degree. Of course, UK Biobank's already considered the business of biomarkers in peripheral blood, and the centralized biomarker panel for all 500,000 UK Biobank participants has already been talked about today as well, and it was released about 2 months ago. And I took this slide from a Naomi Allen presentation. Essentially, the strategy for inclusion was established risk factors for disease, diagnostic markers. There's a protein there. Obviously, there's lipids, there's sex hormones. And then when we get down to characterizing phenotypes not otherwise assessed, biomarkers for renal and liver function, we'll find small molecules. And I think maybe some proteins. Yes, there are some proteins. So in respect of liver, for example, you do have some proteins down here. And diabetes, you've got hemoglobin there, proteins over here. And then you've got, obviously, with respect to cardiovascular, you've got cholesterol, et cetera, and then we've got some hormones down here. So these are what I'm going to call the known knowns. There's more than 200 biomarkers that you can measure in peripheral blood, and they're not measured in the UK Biobank so far. So those are the known unknowns, and that takes us to Donald Rumsfeld. So we have known knowns, that's been done. What we need is these kinds of omics technologies to take us to not only the known unknowns, which we can measure, I'll show you those using this kind of omic platform, but we can also move into the realms of the unknown unknowns, biomarkers that we didn't know about that potentially offer us benefit in terms of understanding wellness and disease. So we had a workshop to consider proteomics and metabolomics platforms in respect of the UK Biobank and the huge analytical challenge that it throws up because of the size of the thing. What we concluded, these are the conclusions that came out, was high throughput proteomics and metabolomics assays at the scale of UK Biobank are challenging, but they're tractable. We can do them. The approaches are mature, and the costs are not necessarily going to come down much further. I actually think proteomics costs might come down a little bit. So you've heard in the previous talks about metabolomics, and we can say without a shadow of a doubt, given the presentations from the two previous speakers, that we do have a mature platform with the ability to undertake hundreds of thousands of samples in respect of assay size. But the important thing is the conjunction of existing detailed phenotyping and genetic data with the plasma-derived proteomic and metabolomic data. So any venture in respect of proteomics, metabolomics is obviously going to have to link in with all of this data. And again, we've heard about the whole exome characterization. This paper has been mentioned already, 49,960 individuals. So this then takes us to the point where we can consider doing proteogenomics. DNA makes RNA makes protein is the dogma, and I can tell you, as someone who's fallen into the field of proteomics, it's not as simple as that. And in that sense, looking for proteotypes, looking for post-translational modifications, etc., I'm afraid that you need to get your hands dirty and actually look at the proteins. So this is something that we need to do, and we can do it with proteogenomics. We're already doing proteogenomics, especially around the area of oncology, looking at tumors and normal tissue. So that's the way that we need to go. Has this ever been done before? Is it going to yield anything of value? Well, I think this is a paradigm study that comes out of the Institute of Systems Biology, the work of Lee Hood and colleagues, that tells us that this really is worthwhile. So I obviously copied this out of the paper, and I'm sorry it's not that big, but basically it says up here, genetics, it says proteomics there, and it says metabolomics there. So they've done three of the things that are proposed to be done within UK Biobank. Obviously, the genetics has been done. And they've got gut microbiome in there as well. So this is undertaking the kind of omics studies. Over here at the top there, it's clinical data, which is cardiovascular, diabetes. A whole raft of inflammatory markers are measured there in the third box, like interleukin-6, interleukin-8, nutrition, and toxins have been measured. So in looking at this paper and looking at the kind of conclusions that they can generate, then I think we can say very clearly that there is value in combining these kinds of data sets. And if you look at the quote that I've put on the right, then connectivity within analyte communities will be established, and that will enable the identification of candidate biomarkers, new biomarkers. And here we get into the business, of course, of algorithms combining measurement of several different entities in order to understand wellness and understand ill health. And of course, that kind of work is something that can be taken forward with the huge data sets that are available in respect of omics technologies. So I put this example in because it shows that these new kind of approaches are actually beginning to take place. So this is an exciting new technique which is called topological data analysis. It's actually not that new, but in terms of application to omics studies, it is new. So this is a straightforward set of questions about stratification of asthma phenotypes. And what the investigators did, based in part in Southampton, was to look at salivary proteomics. So in that sense, they obtained measurements of a whole range of different proteins, and then using this kind of topological data analysis here, after having performed the mass spectrometry on the proteins here, you end up with the important findings down here, which is you can subphenotype this set of patients, this 246 patients, for specific treatments. So when we get these kinds of data sets, these are the approaches that must be employed in order to extract data of information of value. And what is so marvelous about UK Biobank is the business of the integrated data resource opportunity. So if you look at attempts to unify different bits of data that you can find in the literature, then quite often it's incredibly difficult for the investigator to pull together all of the data to make meaningful conclusions, and it takes a lot of work on the part of their co-workers. in respect of what we can do here with omics and UK Biobank is we can take this data, we can take the phenotypic data where possible, and we can take trajectories, and that gives us this smart data which enables biomarker discovery to a far greater degree because of the sheer size of the cohort. So I'm going to tell you about proteomics now. Proteomics is going to be, will be performed on plasma, I think, as the first entity. And what one has to do is deplete major proteins, then trypsinize your proteins, and then you run them online onto a mass spectrometer. And this technique that we propose to use is called swath MS. And you can see from what's happening here that we're cutting swathes through the eluate that's coming off the liquid chromatography. Essentially, we are measuring the peptide mass in each of these windows, and we are measuring the fragments generated from each of those peptides. So that's quite a big mess of data. One of the things that you can do with this kind of approach is build in reference standards, which we do. They're called index retention time peptides. But you can also build in standards for any kind of biomarker that you're specifically interested in. So what we would propose is that a set of known biomarkers would have heavy label peptides of known molar quantity included in this particular set of experiments so that we get absolute quantification on known biomarkers. Then you essentially can build what's called a swath map from all of this data, from the fragments, from the peptide mass-to-charge ratios, and because you've got standards in there, you've got a systematic, unbiased methodology for creating a digitized proteomic map of plasma proteins. And as I said, we only need a small sample volume. And the pre-prepared reference libraries that you interrogate these maps with give you the identity and the relative quantity of hundreds of proteins. And that's one of the reasons why doing mass spectrometry is a cheap approach to addressing the issues around UK Biobank. It's good use of sample because such a tiny amount of material gives you so much information on so many proteins. So the metabolomics has already been talked about in terms of methodology, so I'm not going to dwell on this. In effect, with the proteomics, we're going to be able to identify active players in the field. Obviously, DNA is the blueprint and proteins are the structure, so to speak. And the metabolites also will give us information about the structure and function of the organism. So I've made the point that proteomics cannot be an island unto itself, metabolomics cannot be an island unto itself, and there has to be a marriage of all of these different data sets if these omics technologies are going to be a part of the overall strategy for UK Biobank. And I've made the point about proteogenomics being an important area to be explored. This then, so we have attempted to make a synthesis here on behalf of UK Biobank by developing a British Heart Foundation Bigby challenge bid, which in part has enabled us to think through strategy to a greater degree. So when we acquire data, as we hope to do, then it has to go into an artificial intelligence and machine learning environment and be used for things like topological data analysis. And then out of that will come predictors of risk, the ability to identify subgroups, and potentially trajectories and points for intervention. And obviously it says cardiac here because we made it a bid to the BHF. The important thing is that we end up with point-of-care tests which can actually be used to take clinical decisions as well. That would be a great outcome. And because we want all that, we built a consortium to undertake that work. And the consortium has to start with measurement, so that's proteomics, where you would have Cedars-Sinai in Los Angeles, Manchester, and this company, SciEx, which is part of the Danaher Corporation, which would work together to develop the proteomic profiling. We want to marry all that to the cardiac imaging and brain imaging, especially around vascular dementia or potential vascular dementia. And then we go to Cambridge, Oxford, Stanford to look at data integration. So it's a shame we couldn't hear from Russ Altman because he agreed to be part of this consortium. And from there, hopefully, one would move to usage of the data for point-of-care testing. So I have finished, and later I'll be happy to take any questions. Thanks for your attention.